Thank you, Plante, for the opportunity to present a short talk. My name is Derek Lundberg, and I'm a postdoc in the Weigel Lab, Tübingen, Germany.
And my name is Pratchaya. I have finished my master program and now work in the Weigel Lab. So in nature, as you know, that the plants differ in microbial load, and each color represents the different types of the microbes. As you can see here, the purple and yellow, see they have the same amounts of the microbe across these 5 plants, but the blue is increasing. And here is what people usually do, is that to use 16S RNA amplicon sequencing to observe the microbial abundance. And by doing that, they calculate the parameter called compositional abundance or relative abundance that scale everything up to 100%. And here is a problem. When you look at the sample 5, the yellow and purple have fewer amounts in comparison to sample 1, which leads us to misinterpret and calculate the false microbial abundance.
This problem of compositional abundance is well known and requires special statistics. However, none of these statistical methods can tell you for sure what is actually happening in terms of which microbes are decreasing or which are increasing across samples. For this, you need real experimental data on microbial load. One way to get this data is to use spike in DNA. For example, a synthetic 16S gene added based on the sample mass or area. To do that, you have to first measure the sample mass or area, which has measurement errors, and pipette in the spike in, which has pipetting errors associated. Another option is quantitative PCR, but then you have to set up a separate reaction, which uses sometimes more than double the sample. Also has pipetting errors, which add noise, and it can quantify off-target amplicons, like plastids. You can also use colony counting, but this requires fresh sample. It's vulnerable to pipetting errors, and only culturable microbes can be quantified. Finally, it's a lot of work.
So potential solution that we could use is that whole metagenomic sequencing to get all the information from microbes and also from plants. But in reality, we get all the information from plants, and that is the case for the Arabidopsis, that we get around 95% of the total reads that become wasteful. And it's even more wasteful when we work on the bigger plant genome, like wheat, that has around 16 gigabits.
So what Pratchaya and I have done is create hamPCR, which is host-associated microbe PCR. It's a 2-step amplicon sequencing protocol. In the first step, which is 2 cycles, called the tagging step, we have 2 primer pairs, or 4 primers in the same tube. 2 specific to the host and 2 specific to the microbe. These primers add the same forward universal overhang and the same reverse universal overhang. That means that when we go to the PCR step of 30 cycles, a single primer pair, which can bind to these universal overhangs, can now amplify these diverse templates using the same annealing sequence, therefore without bias.
And that is the case for the hamPCR when we get 2 amplicon types. So the first type is for host, and the second type is for the microbes. And that way we will be able to calculate the microbial abundance based on the normalized or equal host amount.
Furthermore, because we use a single copy host gene, the amount of plant DNA is manageable.
And with that, we can apply the hamPCR with 3 primers. So the first primer is for host, and the rest is for bacteria, and in this case, we apply to oomycete.
So we did a simple experiment where we infected Arabidopsis thaliana with Pseudomonas syringae DC3000, a bacterial pathogen, and Hyloparanospora arabidopsidus, an oomycete pathogen. And we infected wild-type plants and immunocompromised plants where we expect the oomycete to grow. When we look at the relative abundance of only the 16S, first of all, we don't see any oomycete because the 16S primers can't amplify them. And next, we see, yes, the bacterial pathogen, but we don't see a difference between the wild type and the immunocompromised plants. In the uninfected plants, we see a diversity of bacterial families. Using hamPCR, which takes into account the host abundance, we see a very different story. First, we can see the oomycete because we use an additional primer pair to detect it. We also see the bacterial pathogen. We clearly see the immunocompromised plants have higher microbial load. Most strikingly, we see that the uninfected plants have extremely low microbial load. And therefore, you can see that on the top, when you scale everything to 100%, it looks like the abundance of some of these families is much higher than it actually is. So in our paper, we have compared hamPCR with some trusted methods, with colony counting, with quantitative PCR, and the results are comparable. We also used whole metagenome sequencing to calculate the load and correlated that with our hamPCR results. And you can see we have a good correlation.
So in summary, the host-associated microbe PCR or hamPCR can be used to investigate the colonization and the infection of the crop plants, including other plants, and also it can apply to bees and other insects. And that can also be used to monitor the cryptic or symptomless infections, and also can track for the quantitative mixed infection study.
Also, it is useful for simpler statistical and network analysis of microbiome data. So we published this and many more details in our preprints on bioRxiv. Please check it out. So again, I am Derek Lundberg, and this is Pratchaya, and we like to thank you